lined up for the beginning of this afternoon. So we're going to go ahead and start off with Adarsha.
Good afternoon, ladies and gentlemen. My name is Adarsha and I've been working under the direct supervision of Mark Meadows and Dr. Ebony Zuyamo's lab at the Center for Neuroscience, UC Davis. My research this past summer has focused on the understanding the effects of the inflammatory response on the spinal cord, and then the testing of a drug in hopes to manipulate the inflammatory response to expedite the recovery of the spinal cord. So this is so the first part I just talked about was here, and the second part here. Okay, so the first part of my research, as I just discussed, was trying to understand more about the inflammatory response and its effects on the spinal cord. And so the main part of the inflammatory response that we're focusing on for this experiment was cell communications, so in this case, cytokine levels. And so our main objectives were to design a spinal cord injury model so that we could actually injure the mice and initiate the inflammatory response, so then we could then take blood samples and actually study the levels of cytokines. So as you can see from this diagram, after the initial impact, there is glial scarring, and this is our main issue. We actually want to downregulate the inflammatory response so that there's less glial scarring, because the glial scarring actually inhibits axon outgrowth and full recovery and cell regeneration. So as I stated earlier, the immediate goals were a controlled and reproducible spinal cord injury experimental design, and then with that we could make a profile or characterize different activities of cytokines after the spinal cord injury. So the experimental design. So first the mice actually underwent a surgery in which their spinal cord was directly exposed and injured by a machine. So as you can see here, this came down and impacted the spine so that we could actually initiate an inflammatory response. And then the second part, and then next, blood samples were taken at the different times shown in the diagram, and sent for blood analyzations for those cytokine levels. And the third part of this project was the mice underwent a transcardial perfusion, in which their entire body was flushed out and then filled with PFA so that the tissue could be preserved, the proteins cross-linked, and further tissue development and analyzation could occur. And so these are the results from the cytokines, the blood analyzation cytokine levels. And as you can see from the graph, IL-6 is the only cytokine that actually shows an apparent influx compared to the sham. And so now we'll go through a series of comparisons of the IL-6 to all the other cytokines that were tested. And so what we can conclude from this research and these results is that IL-6, the cytokine IL-6 actually only is committed to activity in response to inflammation. So this can actually guide scientists to focus on this cytokine and help reduce glial scarring. And so the second part of my research was in vivo experimentation, in which, as I discussed earlier, I was testing the different concentrations of the drug on a neuronal cell culture. And so this is the same picture from earlier, but there's actually the treatment here. And this part of my research is focusing on treatment number 2, which is blocking the inhibition of axonal growth. So hopefully reducing the glial scar so that axon can grow around it and you'll have full recovery. So a little background knowledge on the inflammatory process. So the previous project was focusing on cell communication, and this is more on the pathways that occur in the inflammatory response. So during the inflammatory response, the body actually initiates many different chemical pathways, and the main one we're focused on is the hydration of EETs. EETs are epoxycosationic acid, and this is very attractive to researchers because of their active role in the cell regeneration process. However, the main issue researchers run into is the hydration of EETs into DHETs, which are dihydroxycosationic acids. And they are not as active, and they're almost dormant in the cell regeneration process, and they're very high concentration in the actual forming of the glial scar, which inhibits axon outgrowth. And so studies at the University of Oregon in 2011, Department of Neurosurgery, actually showed that soluble epoxide hydrolase was the enzyme that degrades EETs into DHETs. So what we used in this experiment was an anti-inflammatory drug that inhibits soluble epoxide hydrolase from converting EETs into DHETs in the hope that we will have a higher number of active cell regeneration factors and a lower number of dormant cell regeneration factors. And so the experimental design Over the course of the 6 weeks here, the research I'm doing right now hasn't exactly been done before in this lab, so a lot of protocols and new things were developed. And so, the main part was the culturing of neurons and glial cells in the spinal cord. So the spinal cord was extracted from the mouth, the tissue was then digested and cultured, and then the drugs were added at different concentrations, immunostaining, and then the morphometric analysis. And so here's like a little diagram. As I just said, the spine was isolated, or it was dissected, it was isolated. Tissue from here was then cultured, or digested and cultured into plates, and then the different concentrations of the drug were added to see the effects of hopefully what we want. And so our hypothesis is, as I stated earlier, that decreasing the number of EETs being converted into DAPTs will hopefully promote axon outgrowth, as we are increasing the number of active cell regeneration factors and decreasing the number of dormant cell regeneration factors. So the results in neuronal culture, I actually don't have too much quantitative data yet. This project is actually going to be continued and replicated more, but I do have representative images of the neurons and glial cells from my culture. And so this is an image from the control. So no drug was added, just regular media. And 2J1 is the staining here. This was staining 2J1. This is GFAG, and this was DAPI, and this is the co-localization of all of them. So the 2J1 mainly stains for neurons, and the GFAG mainly stains for glial cells, but because glial cells are also neuronal cells, they can be, the GFAG can stain for neurons sometimes. So this is a picture of the neurons to control. These are pictures from the glial cells to control, so the same whole immunostaining process. And these are the neurons from the 0.001 micromolar concentration. So the three different concentrations of the drug that were added to the wells of the culture were 0.001 micromolar, 0.01 micromolar, and 0.1 micromolar. So this is the lowest concentration, 0.001 micromolar. And as you can see, if you just remember the 211 micrometers from this, and in the control it's only 28. So we do have a large amount of axonal outgrowth from the representative images. And our next concentration was, or these are actually glial cells, sorry, from the same lowest concentration of 0.001 micromolar. And then we have more neurons from the next concentration, which is 0.01. And as you can see, the axonal outgrowth is not as large, but it's still significant compared to control. And as you can see here, the DAPI staining is more significant compared to this last one. So DAPI staining is for the staining of the nucleus. So as you can see, the cross localization here, you can see that the DAPI staining works for the nucleus for the neuron. And then these are glial cells from the same 0.01 concentration. And then we have neurons, and these are the neurons from the 0.1. So our highest concentration of the drug, 0.1. We actually had a little less compared to the other two concentrations. And then this is glial cells from that last, the 0.0, or the 0.1, so our highest concentration. So discussion, like I said earlier, a lot of the analysis and cell counting is still being done. The whole process can take up to a month. It's really it just takes a long time. But the representative images do suggest, as you can tell by the axonal length, it was very large compared, or the difference was very large in the lower concentrations compared to the higher concentrations. So this actually suggests that the drug may be more effective at lower concentrations. And if we're able to use this, if after we get more of our data to confirm that this drug does indeed increase and promote axonal outgrowth, this could be used as a tool to increase the cell regeneration in the spinal cord after injury and help promote the axons to grow over the glial scar so that full recovery can occur. And my acknowledgments, my main postdoc that I did research under was Mark Meadows, and another person that helped us in the lab, Victor Wong. And then my PI was Dr. Ebenezer Yamoa, and then the Young Scholars Program. And then